Good morning and welcome to the inaugural webinar of Chiba's International Visiting Lecture Series. My name is Praminder Sachdev. I'm co-director of the Center of Healthy Brain Aging at the University of New South Wales in Sydney. I would first begin by acknowledging the Bedigal people who are the traditional custodians of the land on which our center is located and pay my respect to their elders, past, present, and emerging. So we are honored today to host Professor Hanzhang Lu, who's here. You can see him alongside me. He's professor of radiology and chief of the neurofunction section at the Johns Hopkins University in Baltimore, USA. His research is focused on understanding brain physiology and pathophysiology using non-invasive MRI techniques, with a special emphasis on their relationship to brain imaging, brain aging, cerebrovascular disease, and cognitive impairment. Professor Lu is a fellow of the International Society of Executive Residents in Medicine, fellow of the American Institute of Medical and Biological Engineering, and distinguished investigator of the Academy for Radiology and Biomedical Imaging Research. He's published over 180 peer-reviewed journal papers and has an age index of 53, total citation over 11,000. And I must add that he also leads the imaging component of the MARC-VCD, big initiative from the National Institute of Health, which is looking at imaging biomarkers of vascular cognitive disorders. So Professor Lu is therefore the right person to speak on brain vascular function, aging, and cognitive impairment. And following his talk, there will be a live Q&A with Professor Lu. So at the bottom of your screen, you will see that Q&A button. As the talk proceeds, you can direct your questions through that Q&A, type them in, and I'll be screening them as well as Professor Lu, and we will respond to the majority of the questions, as much time as we have. We must remember that today is Remembrance Day, so we have to finish at 11 a.m. for a 2-minute silence in remembrance. So we have to be fairly punctual at the time that we end. So thank you everyone for attending from around the world, and now I welcome Professor Hanzhang Lu.
Hello everyone. Good evening from my time and good morning to your time. My time here is about 6 o'clock in the US East Coast. And thank you for this opportunity to present some of our work. And I look forward to more interesting discussion after the presentation.
Thank you. We'll start with the presentation now.
Thank you for the opportunity to present some of our recent work. So today I will be talking to you about vascular function of the brain in aging and cognitive impairment. I have no conflict of interest to declare. So why do we care about vascular function in the context of aging? First of all, there is increased risk associated with age. We can see that this is data from the NINDS representing, I think, United States population. You can see that as we increase from our age increase from 50s to the 80s, you can see that the probability of having a stroke increase, not just linear, but also actually increase. The rate of it also increase with age. So essentially, by the age of 80s, every 10 years we have about a quarter of chance to have a stroke. And then there's also small vessel disease. Shown here is work from Julie Schneider reporting sort of autopsy data from a group of patients who were diagnosed with dementia when they were alive. You can see that there are, even though these patients were all diagnosed with dementia, they all have different pathological underpinnings. Some of them are related to pure pathology, such as Alzheimer's and vascular and Parkinson's disease and Lewy body, etc. But many have mixed pathology. Now, there's no doubt that the Alzheimer is the most common cause of dementia, but if you add up all the vascular related cases, they account for at least half of the population that's studied here. So that tells us that even for small vessel disease, it can be a major component of cognitive impairment and dementia. So what do we know about vascular change in aging? Well, shown here are some of the typical neuroimaging findings in vascular dysfunction in aging. People have been using diffusion tensor imaging to look at infarction, and there's also Y-max hyperintensity shown here in these bright regions, and there's also lacunes. These dark hole indicate that they're essentially dead tissue there. And people also found that when patients have small vessel disease, these space, so-called perivascular space, is increased, indicating that there's small vessel disease there. And also there's small hemorrhage shown here in these dark dots. Well, all these are very important and useful, but they really don't reflect the vascular function itself. They are more of a consequence of vascular damage. And by that time, there's very little one can do to do intervention or treatment. Essentially, they are irreversible changes. So it is important to have a better understanding of vascular function, hopefully at a stage where things are still reversible. So why is this important? Well, let's first talk about the brain. The brain is about 2% of the total body weight, but consumes about 20% of our energy consumption. And we also know that most of the energy budget of the brain is used to support neuroactivity, either spontaneous neuroactivity or evoked neuroactivity. So vascular, obviously, they provide blood supply to the brain and provide energy source to the brain. So it's important to understand sort of this kind of homeostasis of the vascular blood supply and consumption. And I use a cartoon like this to represent this relationship. We know that the blood from the arterial side is almost fully oxygenated and deliver blood and oxygen and glucose to the brain. And at the tissue capillary interface, part of the oxygen that's carried by the blood will be extracted by the brain tissue and consumed by neurons shown here in this yellow cartoon. And the remaining oxygen would go to the vein and be drained back to the heart. So if you use a sort of equation format to represent this relationship, we have this kind of equation. We have blood flow, cerebral blood flow shown here. And we also have something called oxygen extraction fraction, which essentially is the difference between arterial oxygenation and venous oxygenation. So that difference is called oxygen extraction fraction. So that's also important parameter in terms of the vascular function. And then their product tells us how much oxygen has been used by the brain, and we call it CMR2, representing cerebral metabolic rate of oxygen. So this, we can consider this as a homeostasis. CMR2 represents the oxygen consumption or demand. Blood flow represents the oxygen supply or blood supply, or oxygen extraction fraction, OEF, represents the balance between demand and supply. So let's first look at how the blood supply to our brain will change as we get older. Shown here is some cross-sectional data. We acquired 200 some participants ranging from 20 to 80 some years old. This is using MRI measurement of arterial spin labeling to measure their blood flow. You can see that from the intensity of the image, you can see that as we get older, the blood supply to the brain clearly decreases. And that's sort of well known. And more recently, we have done a longitudinal study of sort of this lifespan cohort. We recruited 115 participants from the Dallas Lifespan Brain Study, and they range from 20 to 80 some years old. And the follow-up time was 9.2 years. So we can first see that the blood flow is decreased over these 9 years of follow-up in almost all the brain regions. Essentially, all the brain regions shows a decrease in blood flow. And if you do a more statistical analysis to these data, which is shown here, we used a linear mixed model effect, a mixed effect model, to look at both the cross-sectional effect of age on CBF shown here, as well as a longitudinal effect on the CBF. And we also added some interaction term. It's a crowded slide, but let me try to point to some important findings from this slide. One is that if you look at it, we find that the longitudinal rate of CBF decline is faster than the cross-sectional. This is sort of more or less has been observed in brain volumetric study, in the sense that when you look at longitudinal, the rate tend to be steeper than the cross-sectional findings. One possible explanation is that when you look at cross-sectional, you are really looking at sort of typical young adults versus supernormal older adults, because by the time they are in their 80s, the fact they can come to your MRI scanner to participate in the research studies, that means they are probably supernormal. So there's cross-sectional studies may contain some sampling bias, and the longitudinal study may be better able to depict the change in blood flow. And in this case, you can see about 0.79 ml per angiogram tissue per minute per year. So longitudinal declines faster than cross-sectional. The second point to make is that we see that female has slightly higher blood flow compared to male. The difference is about 3.7 ml per angiogram tissue per minute. And the final point to make in this slide is that we also find an interesting age by time interval interaction. Now, given that the time interval slope itself is negative, that means that when we get older, the slope becomes less negative. So this tells us that the longitudinal change of CBF with age is actually become not as fast in older compared to young. That is to say, younger people actually their blood flow decreases faster than older people. So this is sort of quite interesting, and it's certainly different from the brain volumetric study, where people have found that brain volume declines faster, shrinks faster in older age compared to young. So this is more longitudinal data. You can also plot this same kind of sort of plot using cross-sectional data, where on the left side, the red curve, we are showing how blood flow changes age decade by decade, and on the right-hand axis, we show in the black curve how brain volume changes decade by decade. You can see that in this case, we plotted the beginning time point and end time point to be same, just to show that their cause is different. In that, again, in the brain volume case, it's decreased slower in younger age and faster in older age, whereas in blood flow, actually even in young, in 20s, 30s, the decrease actually is faster, and actually toward the later age, it decreases actually slower. So essentially, consistent with the longitudinal findings shown in the previous slide, that it seemed like in younger people, blood flow actually decreased already pretty dramatically, and actually slow down its decrease in the older age. So that's about sort of temporal pattern. How about spatial pattern? Shown here are the rate of change of blood flow for different major regions. We look at the major brain lobes, as well as deep subcortical region and the limbic region. We found that the rate of change decrease in blood flow is fastest in the deep gray matter, and this is consistent with the notion that in small vessel disease, they most affect subcortical regions. So those subcortical regions are the area where a small vessel is most pronounced. And indeed, their blood flow seem to decrease fastest in this region. In terms of major lobes, cortical lobes, temporal lobes seem to have the fastest rate of decline, and occipital lobe, not surprisingly, it seems to be most well preserved. On the right panel is the brain volume change per year. You can see that in that case, the pattern is certainly different from the blood flow change. In that parietal lobe seem to have a faster rate, and the deeper region is brain region is the second. We also look at between left hemisphere and right hemisphere. How does blood flow decrease with age? Do they have the similar rate? Well, to our surprise, we see quite some hemispheric asymmetry in the rate of blood flow decrease. We found that in frontal lobe and the parietal lobe, the rate of decrease is faster in left compared to right hemisphere, whereas in occipital lobe and occipital lobe, we basically find that the rate of change is faster in the right hemisphere. We still don't know what's the reason, but it seems like there's a sort of anterior to posterior pattern in terms of the rate of CPF decline, in that more anterior part of the brain, the left hemisphere blood flow decreases faster, whereas in the posterior part, the left hemisphere blood flow decreases slower than right hemisphere. Again, we still don't know what's the reason for that. We look at whether handedness would explain this. We found that it did not explain this pattern, the handedness. And shown on the right panel, just for comparison, the brain volume atrophy rate. There's not much of difference between hemispheric difference. Only occipital showed a effect, but for the most part of the brain, there's no real difference in brain volume atrophy. Now, does blood flow matter in terms of cognition? In this 4-year follow-up study, we found that blood flow measured 4 years earlier can predict episodic memory measured 4 years later. And in this case, frontal lobe had the most effective, most sensitive predictive value in terms of episodic memory. And if you do if you ask which part of the frontal lobe that can predict better, interestingly we found it's more sort of the anterior cingulate area that can better predict episodic memory in this 4-year follow-up study. So anterior cingulate is actually quite interesting area according to our finding. This is one of them that indicates that anterior cingulate cortex is important in the context of vascular aging. We also did a study in which we did a 1-year aerobic exercise study of MCI patients. We had 15 MCI patients enrolled in a 1-year aerobic exercise group, and another 15 MCI served as a control group who only did stretching and toning exercising. And just to show that this group MCI is typical MCI, we show that cross-sectionally these MCI patients shown a decrease, showing a lower blood flow in the posterior cingulate cortex, similar to previous findings. But the real interesting finding is shown on the bottom here. We found that after 1 year of aerobic exercise, their logical memory score has improved. And then when you ask what's the reason for this kind of logical memory improvement, we found that it's the blood flow increase in this area. Again, it seemed to be the anterior cingulate cortex area where seem to have benefited from the exercise, showed an increase in blood flow, which is correlated with improvement in logical memory. The final example I want to show about the potential role of anterior cingulate cortex in the context of vascular aging shown is here. This is a separate study from what I did what I showed in the previous slide. This is a normal elderly group, cognitively normal elderly. They are not MCI, they are elderly group, but cognitively they are normal. And in this we did much shorter time, only 12 weeks of aerobic exercise. Without going into detail, I just want to make the point is that these areas, again, it's the anterior cingulate cortex area in this region that showed an increase in blood flow before and after this aerobic exercise intervention. Now, I've sort of told you about how the blood supply to our brain will change as we get older. Let's now talk about the oxygen injection fraction. As a reminder, oxygen injection fraction represents arteriovenous difference in their oxygen content. So let's look at this. Essentially, this index reflects the balance between oxygen demand and oxygen supply. Okay, let's look at how this parameter would change, because it would indicate how our brain is managing this oxygen homeostasis. We did a cross-section study in 232 participants aged from 20 to 80 some years old. Shown here is the OEF value, decade by decade OEF value for these participants. We can see that OEF increases when we get older, meaning that for each unit of blood coming into our brain, we extract a higher fraction of them when we get older. And if you look at the relationship that I just described previously, this is somewhat expected. That is, since blood flow goes down, if we assume that the CMR2 maintains the same across age, then OEF should indeed go up. And we do indeed believe that the increase in OEF is largely due to a decrease in blood supply. However, let's also look at how the brain's metabolic rate changes with age. In the same group of participants, we measured their CMR2, which is the brain metabolic rate of oxygen per unit tissue. Again, we are not talking about atrophy here. We're talking about the unit tissue's oxygen consumption rate. And in this index, we found that actually it goes up with age. Female has slightly higher metabolic rate than male, but in general, with age, it goes up. This is somewhat paradoxical because previous PET studies using, let's say, glucose metabolism have suggested that the brain's metabolic rate would go down, not going up. So this is indeed somewhat controversial. We did do a follow-up study on the same topic in another 118 participants also, and we found that it again showed a age increase in CMR2, although the p-value is always in the 0.01, 0.02 range, so it's sort of not a strong effect, but nonetheless, in two different studies, we seem to find that the brain's metabolic rate goes up with age. We also sort of showed this in rodent models. This is mice whom we've followed from 3 months old to 20 months old, so it's a longitudinal study, and we essentially do a MRI on them every month to measure the oxygen fraction and the CMR2. Again, OEF increase is quite convincingly increased. That's sort of it's clear. CMR2 in this data is also quite significant, which sort of support the human data that brain metabolic may be indeed going up as we get older. Although this is in mice, so mice brains, their sort of lifespan is different from the human brain, obviously. But nonetheless, this provides some additional support that maybe our brain's metabolic rate does go up. Now, what I so far all said all these things about OEF and CMR2 is about normal aging, but what happens to sort of impairment or Alzheimer's cases. This is a group of amnestic MCI participants. We measured their OEF and CMR2. So in these patients, what we found is that actually OEF goes down when you have MCI, and CMR2 also goes down with MCI. So indicating that as we get older, the brains, as we have developed impairment or dementia, the brain's metabolic rate is less, and therefore they extract less oxygen from the incoming bloodstream. So this is sort of consistent with the notion that in MCI and in AD, the metabolic rate is indeed lower. So I should mention that this kind of pattern is is somewhat different from the aging, in that in aging we found that OEF goes up, whereas in impairment the OEF goes down. And we think this is because when we in the aging process, I think the brain is still using its compensatory process, try to work harder, try to extract more oxygen for its daily function. But then when this compensatory capacity is used up, that's when the brain's metabolism, oxygen extraction can start to go down, because essentially it has run out of reserve, run out of compensation at that point. So again, when someone has impairment and dementia, we expect that their brain metabolic rate would go down. So this is sort of MCI impaired individual. What about ApoE, the ApoE genetically carriers? So in this study, these participants are all cognitively still normal. They don't have any impairment. None of them are MCI. But we can already see that ApoE has an impact on the OEF. More specifically, when you have one allele of ApoE 4 and 2 allele of ApoE, it gradually, the OEF gradually goes down, suggesting that even in the preclinical stage, before you have any impairment, the OEF can already show a decrease, meaning that brain's function can really show a subtle decrease in terms of metabolic rate. And OEF can indeed predict executive function in the ApoE4 participants. We also did some mouse model of Alzheimer's study to sort of verify what are we measuring with OEF. And so we did two different type of mouse model of Alzheimer's disease. One is the APPPS model in comparison with a group of wild-type mice. You can see that the OEF showed no difference. In this no difference. But interesting, the brain volume also did not decrease in this group of mice, APPPS mice. And if anything, the brain volume of APPPS mice actually is slightly higher than the age-matched wild types. So indicating that when there's no neurodegeneration, because brain volume is sort of an indicator of neurodegeneration, when there's no neurodegeneration, OEF show no change either. But then when we move to another type of mouse model, this tau model, this particular tau model, we found that OEF showed a decrease similar to the human cases. And interesting, the brain volume also showed a decrease. So this indicates that in mouse model that has neurodegeneration, as shown by brain volume decrease, OEF would indeed go down. So it seems like OEF is a sort of index of neurodegeneration. It can measure neurometabolism and can indicate early sort of neural dysfunction. Now, one final sort of spin of the OEF as a potential marker in aging and cognitive impairment is that we, in a more recent study, what we found is that OEF is also related to vascular risk factors, and its effect, vascular risk factor, has an effect on OEF that's opposite from neurodegeneration. So what we found is that if you just look at a group with low vascular risk factor, sort of more or less relatively pure Alzheimer's kind of a patients group, you can find the same kind of finding that we've reported previously, which is that as patient get impairment or dementia, their OEF goes down because brain is not as active. But we did not find this in the high VIS subgroup. And same thing, you can also divide the patient another way, can divide it into impaired and normal group. We find that within impaired group, depending on their etiology, let's say we can think that the ones with low VIS, they are more due to Alzheimer's pathology, and the ones due to high VIS, they are more likely to do vascular or mixed pathology. You can see that the one due to relatively pure Alzheimer's, they have essentially lower OEF. But for higher VIS group, it's sort of more or less pretty high, actually. It's close to normal. And indeed, when you look at normal subgroup, VIS did not have a big effect on OEF, indicating that when you use OEF as a sort of marker to make diagnosis or make prediction of someone's, let's say, whether someone has Alzheimer's disease, you have to be careful. There is an interaction between VIS and AD pathology. When you look at the low VIS group, then I think OEF is a good marker for neurodegeneration and Alzheimer's disease. But when you come to high VIS group, it's actually it's not that different. And finally, regarding the OEF, just want to show that in the low VIS group, OEF can indeed predict the composite cognitive score, and it cannot predict cognitive score in the high VIS group. And OEF in the low VIS group is related to A beta 42 as measured by CSF samples, and in high VIS it cannot. So again, reinforcing what I was saying, that in the low VIS group, some pure Alzheimer's cases, OEF can predict cognition, amyloid, and other things. So in the next 15 minutes or so, I want to sort of talk about more dynamic vascular function. Because so far what we talk about is more like static. How much oxygen I'm consuming right now, how much blood supply I'm receiving right now in my brain, etc. But we know that the brain's vascular is not a static tissue or static organ. It's changing blood supply all the time because our brain is so dynamic. We are doing different things at different moments. So therefore, the brain's vasculature is also a dynamic structure, a tissue that is the blood flow into different areas is different at different times. So how does the brain regulate blood supply to different area dynamically and in a spatial specific manner? They are done by these small arteries, which is equipped with this sort of ring type of things. They are smooth muscles which controls vasodilation and vasoconstriction of these resistant vessels. Therefore, it can dynamically regulate blood flow. So therefore, the ability of this smooth muscle in the small arteries to regulate blood flow is an important function of vascular function. And this property is called cerebrovascular activity. That is essentially when the brain vessel need to constrict or dilate, are you able to do that? So how to measure this dynamic property? We use CO2 inhalation inside the MRI scanner to measure this property. So essentially, when the person is on MRI scanning, we also alter the inhaled gas content to give a small amount of carbon dioxide in their inhaled gas. And the CO2 is known to be a potent vasodilator, and in normal brain they should dilate the blood vessel and change the blood flow and change oxygenation level in the brain, etc. And that can be measured by MRI. So the idea is that you use the input as a challenge and the measure output in the brain using MRI signals. So indeed, if we do this kind of experiment, this is doing CO2 inhalations, image become brighter. When doing room air inhalation, image become darker. So this kind of brighter, darker cycle is sort of similar to a fMRI cycle, and the only difference is that we are using CO2 as a stimulus rather than using individual stimulation as a stimulus. So by doing that, you can map the brain's cerebrovascular activity on a voxel by voxel basis, which tells you which region has small vessel that can still dilate and which region cannot. So let's look at how does this CVR change with age. You can see that when you look at normal participants in the 20s versus 80s, again, the CVR cerebrovascular activity goes down. It becomes lower, and that's expected because we know that when we get older, our vessel becomes stiffer, we lose some smooth muscle, our endothelium vascular function become not as effective. If you quantify these, you can see that shown in this red curve, from 20s to 80s, the CVR reduced by somewhere around 50%. And this is cross-sectional. This is not even longitudinal. This is cross-sectional data. And in the same period, cross-sectional CBF shows only reduction of blood flow by about 20%. So this already tells us that the CVR is more sensitive to age compared to CBF. And this is because our brain has a function called audio regulation. It tries its best to maintain a constant blood flow. And how does it maintain it? Well, it will have to dilate the blood vessel to attract more blood flow coming into the area. So essentially, in trying to maintain autoregulation, trying to maintain a constant blood flow, the brain, I believe, is sacrificing vascular reserve, sacrificing cerebrovascular activity to do that. And that's why CVR is more sensitive to age compared to blood flow. And we also studied the common sort of vascular risk factor. How does that impact CVR? In this study, we found that compared to normal intensive participants, these are all 55 years and above, hypertension, patients with hypertension tend to have a lower cerebrovascular activity. And interestingly, we also had a group of patients who had hypertension, but after treatment, at present, they are not hypertensive. Their blood pressure is not exceeding the threshold. They are not taking antihypertensives, and for these people, you can see that their CVR is back to normal. So it seems like hypertension does have an impact on the CVR measure. We also did a 4-year follow-up study of CVR to see how longitudinally it would change. You can see that the image is similar between two waves, and they are 4 years apart from each other. So we quantify the different lobes CVR change in terms of their change per year. You can see that the temporal lobe has the fastest change in CVR, and again, occipital seems to be most well preserved in terms of their vascular function, as they show the slowest rate of CVR decline. We also look at which age group would have the fastest decline, and similar to the CBF finding, we found it's not the older group that had the fastest vascular decline. It's the middle age in this case. So it's not the global group, but earlier age. And this can be seen in both temporal lobe and occipital lobe shown here. So again, vascular decline seems to happen early in our lifespan. Unlike brain volume happen late. That is, brain atrophy is accelerated in the advanced age, but actually vascular decline seems to happen in either young or middle age individual. How does this CVR measure relate to the hyperintensity, which is a classic, more classic measure of small vessel dysfunction? We can see that there is a gradual decrease from normal appearing white matter to periregional area in this green area, periregional, and to white matter lesions. There is a gradual decrease in their CVR, indicating that when CVR is down, the brain tissue tend to become hyperintensive, hyperintensive, and can have white matter lesions. And then use this concept, we can say, okay, what if we set a threshold on the CVR map and find which brain region, which tissue have a very low CVR, and we call that high risk CVR, and then we can compare that to hyperintensive volume. And that's what we found in this cohort. This is elderly cohort. We found that the high risk volume of CVR is indeed correlated with hyperintensity volume, even though the sort of this is more anatomical change, this is more functional. So, but they seem to have the same origin, meaning that if someone has lots of high risk tissue in CVR, they are likely to have more hyperintensity in the FLAIR MRI images. So more recently, we participate in a consortium study called the MARC-VCID study. This is funded by the US NINDS, and the goal is to develop biomarkers for vascular contributions to impairment and dementia. And shown here are some of the sites. We have 7 sites that are collecting data. Our institution is one of the sites, and we have a coordinating center headed by Dr. Steve Greenberg from MGH, and again, the NIH NINDS sponsored this sort of fairly large trial. Each site supposedly do a 2-year study to develop their own biomarker, and this is called a UH2 study, and shown here is some of our result. We recruited 28 normal elderly and 44 impaired participants. We purposely had a lot of enriched this cohort with vascular risk factors. We have a high percentage with hypertension, hypercholesterolemia, a decent fraction has diabetes, smoking, and a high BMI. So this is a cohort that's enriched with vascular risk factors because we're interested in vascular biomarkers. We did a CVR scan that's about 7 minutes inside the MRI scanner. When you look at groups, the normal and impaired group, they showed a sort of modest difference, And if you quantify this global CVR, you can find that the impaired group indeed has a lower CVR compared to the normal group. The difference is actually more dramatic when you look at cognitive function. We found that CVR has a very strong association with the global cognitive function indicated by MoCA. You can see that we have a fairly strong association, and this is all correct for age, sex, and education. And this is MoCA. We also did a more comprehensive neuropsych test, and shown here, we found that with a composite score coming from neuropsych test, it was also associated with CVR. And we further also draw CSF from these participants. Therefore, we can look at how CVR and amyloid and tau can simultaneously predict cognition. We found that CVR and amyloid can independently predict MoCA score, and also independently predict the composite neuropsychological score, and indicated that CVR and amyloid, they are two independent mechanisms that can explain the variance of cognitive function in this group of participants. How about specific domains? What we found is that the language domain and to some extent the processing speed domain has the best association, the highest association with CVR. With exact function memory, exact function maybe have a smaller, weak association, but memory has no association with CVR. We also want to compare CVR's ability to predict cognition with hyperintensity and vascular risk scores, because hyperintensity and VIS are sort of more classic measures of small vessel disease. So we put all three, CVR, hyperintensity, and vascular factor, in the same model to see who can better predict cognition. We found that only CVR was able to independently significantly predict cognition, whereas the other two was not able to predict MoCA score. And we found CVR was not associated with physical score or VIS, although as I mentioned previously, CVR was found to be sort of related to hyperintensity volume, but when it comes to physical score, it was not correlated. And we also want to make sure that CVR measure is reproducible in terms of predicting cognition. So therefore, in the same participant that I described above, we also did another type of CVR, in which instead of doing so-called bold MRI imaging, we did a blood flow using phase contrast MRI imaging. You can see that this is an image in the superior sagittal sinus. You can measure them twice during room air breathing and during CO2 breathing, and then their difference tell you the blood flow change. So using this, we call it blood CBF. CVR essentially is a different type of CVR measure. We want to show that with a different measure, we can still predict the same kind of cognitive function. And indeed, shown here is the result. There is a group difference, again, moderate difference between normal and impaired group, but a fairly strong association between CBF-based CVR and the MoCA score. So this seems to indicate that CVR is a quite reproducible measure that can predict cognitive function, and it does not really depending on which specific MRI technique you are using to measure CVR. And this all has been corrected for age, sex, education, amyloid, tau, and all the other things. So it's still quite significant after you account for all these things. Like I said, our MarkVCID project has two phases. UH2 phase is the first 2 years. UH3 is the latter 3 years. So this is what I've shown before, the first 2 years there. More recently, we are collecting the sort of the new data in the UH3 phase. And you can see that in a different cohort, you can see, and then 3 years later also, you can see that the MoCA and CVR is still correlated, and this is after adjusting for age, sex, and education, suggesting that this CVR in terms of predicting cognitive function is quite robust and again quite reproducible. So to summarize, I hope I've shown you that the brain's blood supply decreases with age, and interestingly, it seems to decrease faster in younger than in older individuals. In terms of spatial specificity, subcortical tissue shows the most fastest decline in blood flow, and that's consistent with the notion that small vessel disease affects subcortical tissue more prominently. Oxygen infraction fraction is an interesting index, and according to our current data, we think that it may represent neurodegeneration, at least in the patients who are more related to Alzheimer's disease, more related to amyloid kind of pathology. In those patients, it seems like OEF is a good indicator of neurodegeneration, and potentially can be used as the N indicator in the ATN classification system. But again, it seems to be only working in the low vascular risk factor patients. When you have higher vascular factor, the change in OEF is a bit different. It may not show much change. And I also present a new measure called cerebrovascular activity, which measures the small vessels' ability to dilate, and we think that it's a potential marker for small vessel disease. I would like to thank our funding agency for their generous support, and thank you for your attention.
So thank you so much, Professor Lu. That was a brilliant talk, and congratulations on the excellent work of your center. So as you can see, there are a few questions that have come through on the panel. I don't know whether you can read them. So the first question is from Jiang. I'll just very quickly say what it is. He basically asks you how the metabolic rate for oxygen relates to affects the Bold signal. So
thank you for that question.
Aging versus younger people. Yeah.
Right. So I want to clarify that in terms of technique, well, I've presented in terms of CMR2, CVF, and OEF, they were all measured under resting state. That is, the person was not doing any visual task, motor task, or cognitive tasks. Whereas Bold signal typically is measured under a particular task. So what we are saying is that today I was mainly reporting the basal or resting state metabolism and neural activity, whereas Bold signal, at least in the context of evoked fMRI signal, that primarily reports the sort of task-induced activity. And just very quickly saying that in the cognitive aging literature, I believe the primary finding is that people find in frontal lobe activation, older people have bigger Bold fMRI signal compared to young, and that seems to be consistent with our indication that older people have a higher metabolic rate compared to younger folks.
Okay. On a related issue, I wanted to ask you about techniques, really, because I don't think you went into that in detail. But there are several techniques to measure cerebral blood flow. For example, you have use an exogenous contrast like gadolinium, or you have ASL. What is your preferred technique, do you think, and how do the various techniques compare?
Right. I think the first technique, I'm going to be very honest with you. The first technique I will say don't use is the gadolinium-based dynamic stability contrast MR technique. The reason is that it is primarily used in clinically for, let's say, the case of stroke or brain tumor. But for agent study, I think that's not the best choice because it first involves conscious agent. And second, it's not very quantitative. It does not give you a absolute value in terms of milliliter blood per 100 gram tissue per minute. It's more a relative method. So if you have a stroke region compared to healthy, that's fine. If you have a tumor region compared to a non-tumor, that's fine. But for quantitative measurement in cognitive agent, I would say don't use that. And the other technique is ASL. ASL has advantage of spatial resolution and also does not require conscious agent. So I would highly recommend ASL, arterial spin labeling. And the other technique we've been using in our laboratory is something called phase contrast quantitative flow MRI. The downside of this phase contrast method, it has no spatial information. It only gives you one value per person. Let's say I tell you my brain is 55 mil per 100 gram tissue per minute. That's my blood flow. But I don't know which region has more, which region has less. But the upside is that it's very quantitative and it's fast. It takes less than 1 minute to measure.
Oh, okay. And with ASL, I just follow on. Is there some special protocol in ASL that you recommend over others?
Yes. If you're using ASL, I believe all major MRI vendors, such as the Siemens that your scanner is manufactured by, they have product sequence using sort of standard protocol. So ASL has been a quite standard technique nowadays. It should be accessible to you.
Okay, thank you. So we move on to the next question from Nikki Caution, who's a neuropsychologist in our group. She basically wants to go back to your work on hypertension and CVR, and wants to ask you, how do you explain the rebound, really? I mean, of the recovery of CVR in people with previous hypertension? What was the basis for their hypertension? And also, in terms of effect of medication on CVR, antihypertensive medication on CVR?
Right. My short answer is I don't have a good answer for that. The medication that these patients used varies. This is not a homogeneous clinical trial kind of medication.
Yeah.
So this is just an observation that we found. They seem to help, but given the heterogeneity of the medication they are taking, I don't have a good answer actually.
Oh, okay. And you, in terms of relative different medications, would you say that there is differential effect? Say, for example, ACE inhibitors, say ARB antagonists. So does it matter which drug you're taking?
They have both. In our participants, they have both. So like I said, the ideal thing, which actually we are trying to do with a collaborator, is to use one type of drug to try to see if it has an effect. And the drug that we are trying to do is, what is it called? It's, atorvastatin. It's a statin, basically.
Statin, okay. Yeah, okay. Yeah. All right. So it would be interesting to see what statins do as well.
Right. If that, in that case, it's a pure study. It's a one drug, one medication. Then we can better understand that.
That's true. So that's an interesting question, I think, to follow up in terms of the relative, because there is some evidence that some drugs some antihypertensive may be more protective than others, although the evidence is very mixed. So it may be worth seeing whether what effect they have on CVR. So in related question, I think with CVR is, of course, when Kailash Roberts asked this question, what is the effect of this change we see with aging in CVR and the glymphatic mediated clearance of A beta and tau? Do you think that the two are related in some way?
I think so. I think the ability of the vessel to kind of dilate and constrict, certainly people have said that's the driving force for the lymphatic flow.
Yeah.
So I certainly would imagine that a person who have lower CVR would have slower clearance of waste and potentially a faster buildup of amyloid and tau. No one has tested this, so we don't know if that's true. But I would certainly think that if you want to measure lymphatic system's driving force, CVR will be one of them to study.
Yeah. Okay. There's no direct measure of the lymphatic flow, is there?
I am not aware of sort of a image measure that can be done in human that can measure lymphatic flow, because essentially you have to. Ideally, you have to inject some small tracer into the brain parenchyma, into the interstitial fluid to see how it flows. So I don't think there is a way to measure the actual flow in vivo. But in terms of measuring driving force, I think CVR could be one of the tools to measure driving force. The other tool to measure the driving force is to directly measure the pulsatility of arteries in the brain. And there are imaging methods that can measure that kind of things, measure the arterial pulsation to see if the driving force
Otherwise, one would have to go to an animal model, I guess, to try to relate the two.
Right, you have to do animal model, but animal model, I don't think it's easy either. And I think any in vivo measurement is not trivial in this case.
Yeah. Sure. Okay. No, that's interesting. I think there is this interest in, of course, the change with aerobic exercise. I think that's the interesting finding. And Catherine asked this question, what does it mean, lifespan approach to aerobic exercise, the rate of declining neurodegenerative disorders in old age? So, I mean, I suppose your data support that.
I think so. I mean, as you know, there are a lot of evidence suggesting that aerobic exercise reduce your rate of potentially have dementia. But I think the effect size is pretty small, I think, because it is not like a real medication. You can turn things back. I think it's beneficial, but the effect size may not be that big. And I think our study essentially tells you that what is the mechanism by which aerobic exercise can work in that. So that also means maybe there's a shortcut. If you can magically increase the blood flow to the anterior cingulate cortex, maybe you can amplify that effect.
Yeah. Okay. Let's hope we have some other intervention which can complement exercise to that. Yeah. So one area that I think you didn't touch upon is measuring the blood-brain barrier with imaging techniques. Is that something you're looking at as well?
Right. I did not present that data in this presentation, but yes, within the MR field, there are a few new techniques that can be used to study blood-brain barrier in humans. Yeah. And some use contrast agent, some do not use contrast agent. Which one is better? It's still being debated. Our laboratory is working on one that does not require contrast agent.
Yeah.
Unfortunately, it does not have spatial information, only give you a global measure, but the nice thing is that it does not require contrast agent. And our papers has not been published. That's why I haven't reported it here. But our initial study suggesting that there was a difference between MCI and older elderly normals in terms of their BBB permeability using our method.
Oh, okay.
But there are other techniques that you are all welcome to consider using contrast agent.
Okay. So I have a few more questions. We just have 1 more minute to go, in fact. One is the relationship of CVR with cognition. So the question is, can you clarify, is it largely neural or vascular? Does the ability of neurons or astrocytes to signal for vasodilation change? Or is it parasites that change? What is determining this?
So for sure it's not neural. I think it's vascular. In terms of vascular, what aspect of vasculature, such as whether it's endothelium cell or it's the astrocyte end feet, that we don't know yet. But certainly we don't think it's neural. It's the vascular itself. Whether it's smooth muscle or endothelium or astrocyte, we don't know yet.
Yeah. Okay. Jiang has a technical question. I think maybe you can answer that later. And Ashley Bush, who's a leading dementia researcher in Australia, he asked this question: oxygen itself can induce oxidative damage, and its concentration must be limited to prevent reactions, for example, lipids. So with the increased OEF with aging, is there any evidence that oxidative stress markers are associated?
Let me quickly, as I said, time's up. Oxygen extraction fraction high does not mean the oxygen tension is high. There are 2 different measures. In fact, I believe that the oxygen tension in older is lower.
Okay. Thank you. Thank you very much. We have to finish. Want to thank you again for this excellent talk, and of course all the attendees for logging in and staying with us. And we welcome you to the next lecture on the 9th of December, which is from Professor Ron Peterson from the Mayo Clinic on diagnosing Alzheimer's disease in the biomarker era, promises and pitfalls. Of course, as you know, Ron is a leading researcher in this field and very well known. So look forward to seeing you again. Thank you again, and bye for now.